Good morning, my name is Jeff Petracca. I'm one of the educators and entomologists here at the DNA Learning Center. And today we're going to do one of my favorite labs, the DNA restriction analysis of the lambda phage genome. But before we do that, I want to talk a little bit about DNA. So up here I have a model of DNA. I'm going to untwist it right here. And notice that DNA sort of looks like a ladder. So we have the black and white pieces here, sort of being the ends that you grab onto the ladder, and then these little colorful bits in the middle here that are kind of like the steps on a ladder. The DNA molecule has a structure that's known as the alpha helix, so meaning that this ladder is basically twisted, kind of like a spiral staircase, much like this. Now, DNA is a nucleic acid, and it's made up of building blocks called nucleotides. So on my model here, notice that we have these white and black pieces comprising the backbone. The white piece is known as deoxyribose sugar. The black piece is known as a phosphate, and these colorful pieces in the middle here are known as nitrogenous bases. So together, those three components make up a nucleotide. Nucleotides are monomers, and they create long chains of monomers that are called polymers that eventually create a nucleic acid. These two strands unite together to create the structure of DNA, the double-stranded structure of DNA, that is. Now, what I want you to notice, if you look in the colorful sections in the middle here, you'll notice that certain colors always go with other colors. And that sort of highlights the idea of complementary base pairing. So what I mean by that is there are 4 bases in DNA: adenine, thymine, guanine, and cytosine. A, or adenine, always goes with T. G, or guanine, always goes with C, or cytosine. And so that's pretty important for what we're going to be talking about here today. So DNA is a pretty important molecule. So all living things are made up of DNA. So me, plants, animals, even bacteria contain DNA. So I have a picture up here of a bacterium known as Escherichia coli. It's a bacterium, and I use this picture for a good reason, because we're going to be dealing with these guys a little bit today. And so the reason that DNA is so powerful is because it shows us that all living things are related. So the fact that bacteria have DNA and the fact that a human, something as complex as a human, has DNA, shows us that we're all related. Now, the reason DNA is so important is because it contains the building plans for proteins. So basically everything that is us is all the building plans, the information for building the proteins that make us up, are contained within DNA. So does anyone know what that's called? So if you said gene, you would be correct. So a gene is a specific sequence of nitrogenous bases that contain the building instructions for building a chain of amino acids and then consequently a protein. Okay, so up on the board here, I have what's known as the central dogma of genetics. So the idea that DNA contains information that can be transcribed by RNA, and then that RNA can carry the message of the DNA to the ribosomes, where it can be translated into a chain of amino acids, thus building a protein. So this is a pretty important biology. So now, let's take a closer look at a bacterium. So up here, I have a sort of a model of a bacterium. I think like candy, has little features in there. And what you should notice here is that the DNA of bacteria first of all, it's much less complex than that of an organism, of a eukaryotic cell, such as the cells that make up a human or a plant. And they have just one chromosome sort of floating around in the middle of the cytoplasm in a region called the nucleoid region. I can sort of highlight that right here for you. However, they also can have extra DNA. That DNA can be found on little tiny rings called plasmids. So it's that little sort of candy ring that you see in the middle of the model here. Now, plasmids are pretty important. So what I want to talk about next is how plasmids can be accessories for bacteria. So essentially, when we think about going to the beach, for example, what do you wear when you go to the beach to protect yourself from the sun? Sunglasses, right? So sunscreen, hats, whatever. Those accessories protect us from the sun. Plasmids can act in very much the same way as sunglasses or sunscreen. They can give bacteria an advantage over other bacteria in a particular environment, such as for example, if a bacteria lived in an environment that had an antibiotic present. So you guys may know what an antibiotic is. Antibiotics are essentially chemicals that can either kill or prevent the growth of living microbes, such as bacteria. And so if you're a bacterium and you find yourself living in an environment that has an antibiotic present, you kind of might be thinking like, ooh, I don't know what I'm going to do about this. But if you had a plasmid that contained a gene for antibiotic resistance, for example, you have an accessory gene that can help you survive in that environment. And the other reason that plasmids are so important, it kind of relates to us as humans and genetic engineering. So it's this idea of recombinant DNA technology. Because plasmids are tiny little rings of DNA, we can easily get them up, or relatively easily get them up into other cells. And we can sort of mix and match genes and build a plasmid that we can use to insert into bacteria to make them express a gene that we might be interested in. So, for example, I told you guys I was an entomologist, right? So entomologists study insects and spiders. So I had to include a bug example. I'm sorry if you don't like spiders, but I have to talk about this just because it's so cool. So up on the board here, I have an image of a spider called Nephila clavipes. So that's a big spider right up there. So it's a good like 3 inches across in leg span. It comes from Florida and Central America. And it looks very scary, but just as an aside, it doesn't hurt you. It can't do any damage to you. Don't ever be afraid of the spider. But the cool thing about it is the silk. So as it turns out, spiders in general produce a very, very strong protein silk. It's actually one of the strongest biopolymers out there. And because silk is a protein, where do you suppose we could find the building plans for building silk? You could find them in the spider's DNA. The spider has genes that encode for silk protein. So it's a little more complicated than I'm letting on, but the idea is all of these different colorful boxes down here represent different genes that can encode different types of silk for the spider. So you might be asking yourself, like, really strong biopolymer, that sounds pretty cool. So interested in that. So as it turns out, people are looking at spider silk for various different applications, including medical applications, so nanosurgery, using them as scaffolding for rebuilding damaged cells, also in building material. So recently someone incorporated spider silk into wood cellulose composite and was able to build a much stronger building material. You could also even use it for military applications for like bulletproof vests and things of that nature. So we might be interested in obtaining the building plans for building silk protein. So since those building plans are contained within the genetics of the spider, we need to figure out a way that we can pull those genes out and incorporate them into a little tiny ring of DNA called a recombinant plasmid, making a recombinant plasmid. And then we can try to get that recombinant plasmid up into a bacterial cell through the process of transformation. If you haven't already done so, you should check out the DNALC live video last week with Erin, where she did a bacterial transformation. It's very much the same idea. And so the bacteria will then be able to produce spider silk protein for us. But again, how do we actually get at those genes? That's the question. So that brings me to this sort of next point. So first of all, what are enzymes? Who can tell me what enzymes are? I'm going to answer this question right up here. So enzymes, you guys should know, reduce the activation energy of a particular reaction. They reduce the activation energy of a particular reaction. And a different way of saying that is they make reactions more easy to occur by finding an alternate thermodynamic pathway. So they basically sort of make it so that reactions can occur in a different and more efficient way. Now, enzymes in biology, you may know when you see them written by the last 3 letters, ACE, A-S-E. They have that at the end of their name. So do we have any enzymes that could be used to cut DNA? So yeah, as it turns out, there are enzymes called restriction enzymes that can be used to cut DNA. Okay, now, restriction enzymes are pretty important. Whenever you see that word restriction in biology, you should immediately think to yourself, cutting DNA. So, restriction enzymes, there are hundreds of them, but we might sort of ask ourselves where they might have originated from. And so you can actually find these restriction enzymes in nature in bacterial cells. So it turns out that bacteria use these restriction enzymes in order to protect themselves from viruses. So from specifically bacteriophage viruses. So viruses that like to come and attack, look to seek out and attack bacteria specifically. So let's talk about viruses for a second, because we're actually going to be the lambda-phage viral DNA shortly. So I want to kind of get an idea of, give you guys an idea of what these bacteriophages are and what they look like. So right up here, I have a picture. There's actual pictures of lambda-phage viruses. And they're very small. We're talking like nanoscale. They're very, very small. And so down here on the bottom left, you can see an image of a bacterial cell here that's being infected by multiple different phage virus particles. And notice that these viruses have a very specific structure. So if you look at the top image up here, you'll notice that they have a head called a capsid that contains the genetic material. So in this case, for a lambda-phage virus, there's DNA. So we call that the capsid. Then we have a little protein tail, that's this piece right here, that acts as a sort of syringe to inject DNA into a host cell. And then we have little tail fibers down here. These little tail fibers are essentially kind of like landing machinery that help the virus identify specific target receptors on the surface of a bacterial cell and bind to that cell specifically. And so what you should notice is that both the capsid as well as the tail are made up of protein. However, again, these viruses are nanoscale. They're very tiny. Why do they even have to attack bacteria in the first place? So it turns out they lack the important machinery for building cells. So specifically ribosomes. They lack ribosomes. Those are those units that build protein from RNA. And so because they lack ribosomes, they have to essentially hijack another cell and take it over and make that cell produce more of their protein for them. That's the idea. So therefore, if you're a bacterial cell and you have to constantly worry about these phage viruses coming at you, it would sort of behoove you to have a defense mechanism against those viruses. And that's where restriction enzymes come in. So here's the idea, by the way, with the viral attack. So here we have a little phage virus up at the top of the screen here. E. coli cell. Notice the size differential there. So the little phage virus will land on the surface of the cell, and then it will inject its DNA into the middle of the E. coli cell. And it will try to hijack the bacterial ribosomes to start making more of itself. Just like this. So now we have many little viruses. These viruses, once they're assembled and built, will burst out of the E. coli cell, and they'll go off and start infecting other E. coli cells as well. That's the idea. Okay? So if you're a bacterial cell, how do restriction enzymes help to resist this bacterial virus attack? So what they're going to wind up doing is once the viral DNA is inserted into an E. coli cell, the restriction enzymes will recognize specific sequences of nucleotides, specific cut sites on the viral DNA, and go up and chop it up and break it up so that the viral DNA cannot take over. Pretty cool, right? So for today's experiment, we're going to be talking about two different restriction enzymes. And whenever we do a lab here at the DNA Learning Center, I always like to sort of emphasize what the purpose of the day is supposed to be. So if you're following along at home and carrying out the experiment, or you're going to be coming here in the future, always remember that when you come here to do a restriction analysis, the whole purpose of today's lab is to identify a mystery restriction enzyme. So we basically have a tube that has a question mark on it that is one of these restriction enzymes, but we don't know which one it is. That's the idea. Okay, so let's take a closer look at restriction enzymes. So first of all, we have EcoR1 and we have Hindi3, two restriction enzymes. Notice that they don't have the ACE ending that other restriction enzymes, that other enzymes have. Now that's okay. Restriction enzymes are actually types of endonucleases. They're named for the activity carry out. But don't get harped up on the name. They look pretty complicated and fancy, but they're really not that sort of hard to sort out. So EcoR1, the reason that it's named that way is because it was discovered in the Escherichia coli RY13 strain of bacteria, and it was the first one discovered. So we have the name EcoR and then 1. Hindi3 was discovered in the Haemophilus influenzae RD strain, and it was the third one discovered. So we have the name Hindi3. More importantly, though, these are type 2 restriction enzymes. So there are a lot of different, I told you, there are hundreds of different restriction enzymes. But I want you to take a look at the specific sequences that these restriction enzymes identify and cut at. And I want you to tell me if you can see specific about the way that those sequences are structured. What you should notice is that if you look from for EcoR1, for example, if you look from the 3' to 5' end and the 3' to 5', the complementary 3' to 5' end on the other side, you'll notice that it is the same sequence forward and backwards. So we call this a palindromic sequence. Palindromic sequence, just like from English class, palindrome, a word or a phrase that's spelled the same way forwards and backwards. So these restriction enzymes will identify these specific sequences of nucleotides, and they are going to cut in a very specific way. So it turns out for EcoR1, they cut in between the G and the A on the 3' to 5' and the 5' to 3' sequence. That leaves behind this sort of zigzag cut pattern. And when it does that, it's pretty cool. So it essentially leaves behind fragments that have these sort of overlapping complementary ends that are single-stranded, and we call those sticky ends. And the reason that we call them that is because those two sticky ends are going to want to join back up with each other, undergoing that complementary base pairing that we were talking about earlier. And so, for example, if I started with a fragment of DNA over here on the top left, you will notice that if I took EcoR1 and exposed this little fragment of DNA to that restriction enzyme, it's going to identify the GAA-TTC cut site, and it's going to cut that fragment into 2 pieces, leaving behind 2 fragments. Okay, so how does that play into the genetic engineering component that we were talking about earlier? So back to that spider that we were talking about. So if we needed, so here's that golden orb weaver. Here's an up-close image of it up here on the board. And so if we wanted to get at one of the silk genes that were present in that spider, we would identify that silk gene through its sequence. We would figure out a cut site that would occur on either side of the gene. So for instance, in this example, I noticed that I have a GAA-TTC cut site on either side of the silk gene. So I might want to use enzyme EcoR1 to go ahead and cut that spider's DNA at those locations. That would essentially remove that gene for us. So back up on the board here, I can show you, take that spider's DNA and I expose it to EcoR1. It's going to recognize those 2 cut sites, and it's going to cut out that silk gene. Hang on one sec, sorry guys. Okay. Now, let's say that I wanted to incorporate this into a bacteria or some other piece of DNA. I would then take a plasmid, let's say, and I would expose that plasmid to the same restriction enzyme, essentially cutting at the same exact location. So notice that I have that GC cut site here in that plasmid as well. So EcoR1 is going to cut there, and it's going to break apart that DNA. Then if I mix the 2 pieces of DNA together, oh, hold on, guys, sorry. I wind up essentially being able to combine those pieces of DNA into a recombinant piece of DNA that can be into a bacterium or some other host to express. Pretty cool, right? All right. So let's talk about how we're going to carry out our lab today. So the lambda phage genome, so that's the entire is relatively small. So it's basically about 48,500 base pairs in length. So this is an image up on the board here of a restriction map. So a restriction map essentially is like a reference map that tells researchers where different enzymes will cut various different pieces of DNA. So down on the bottom here, notice that we have the restriction map for EcoR1 and we have the restriction map for HinD3. So basically, in order for us to figure out where these enzymes are going to be cutting, we need to sort of know the sequence. And what we're going to do is expose the lambda genome, so this entire genome, to each of these different restriction enzymes, and we're going to be able to compare that restriction enzyme, the mystery restriction enzyme, in order to figure out which one that we have. So notice that they have a distinct pattern of cutting on the restriction map here, so we'll be able to differentiate them that way. Okay, so let's talk about how we're actually going to do that. So what we need to do is essentially come up with a couple of different what are called restriction digests. So restriction digest, basically taking DNA and digesting it with a restriction enzyme or cutting it up with a restriction enzyme. So in a science lab, one of the helpful tool that we often use are microfuge tubes or microcentrifuge tubes. So up at the front of the room here, I have a couple of these little microcentrifuge tubes. I've got four of them actually. And we're going to basically label these just so that we know exactly what we're going to be putting into each of the different digests. So the labels are going to be basically what you see up here on the board. We have 4 tubes. We need to expose the genome of lambda phage to each of the different restriction enzymes. So therefore we need at least 3 different treatments, but because this is a science experiment, like all good science experiments, we need sort of control. So we're going to also include a control tube, which we'll talk about in a moment. So right now I'm going to go ahead and label these microcentrifuge tubes. So I'm going to label the first one E. I'm going to label the second one H. I'm going to label the third one minus, and the fourth one question mark, just like you see up on the board here. Okay, I'm going to take those tubes and then I'm going to stick them into this little test tube rack right here, just for easy access in a moment. Okay, so what do we need to actually carry out this experiment? So what do you think has to go in all 4 of these tubes? So if we're trying to figure out what our mystery restriction enzyme is, we need to be cutting the same DNA in each of the different digests. So therefore, we need to put lambda DNA in all 4 tubes. That's one component. The next component, we need to add enzymes to each of the different tubes. So basically, our E tube is only going to get our EcoR1 enzyme. Our H tube is only going to get HinD3 enzyme. Our mystery tube is only going to get our mystery enzyme. And then for the minus tube, the minus is to indicate the lack of a restriction enzyme. So instead, what we're going to add is distilled water. So this is going to serve as our control. And the reason that the entire genome is actually intact to begin with, and that's exactly what the minus tube will do for us. Okay, but what else do we need? So when we talk about enzymes, very often we talk about the conditions under which they operate. So there are various different things that can influence the behavior of a particular enzyme. So one of those would be temperature, for example. Another would be pH. So the fluctuations in temperature and pH can wind up creating essentially changing the way that those enzymes behave chemically by altering their shape usually, or their chemistry. And so we need to control for those things. Temperature is not going to be a big deal. So temperature, we are going to essentially carry out this experiment at the same temperature, specifically at 37 degrees Celsius. 37 degrees Celsius should sound sort of familiar to you. And that's because 37 degrees Celsius is the human temperature. So the bacteria that these enzymes originated from, E. coli and H. influenzae, they occur in human bodies. So therefore, it would make sense that their enzymes would perform optimally at 37 degrees Celsius, or human body temperature. Sounds like a good assumption, correct? Yeah. So what about pH, though? How are we going to control for pH? So if you remember from chemistry, we can control for pH using a solution known as a buffer. So a buffer is essentially a solution that resists changes in pH. Okay, so we're going to use that to control for pH across our 4 digest tubes. Okay, so all 4 of these tubes are going to need to get a restriction buffer, just to make sure that all the enzymes are doing their thing in a uniform environment, more or less. Okay, so we're going to be dealing with very small quantities of liquid here today. So the tool that molecular biologists would use to measure small volumes of liquid would be a micropipette. So it just so happens that I have a bunch of micropipettes right here. So at the DNA Learning Center, we use basically 3 different pipettes on a regular basis. They're all different colors, and they each reflect different sizes. So I have a blue pipette here that is the largest pipette. I have a yellow pipette that's sort of like a middle-sized pipette, and then I have a gray pipette that is the smallest one. And we're going to actually be using this pipette. The gray pipette measures anywhere between a half a microliter to 10 microliters. So that prefix micro is pretty important. So micro, so up on the PowerPoint here, it's like a U, but it's really written as like a fancy U, kind of like this. It's called a mu. And that prefix micro is basically telling you that a microliter is a millionth the size of a regular liter. So it's 10 to the negative 6 liters. 1 microliter is that is a millionth the size of a liter. So to put it in perspective for you, if you let a snowflake or something fall on your fingertip and you let the liquid melt, the liquid left behind would be anywhere between 1 and 2 microliters. So it's not a lot of liquid at all. And that's sort of the size range that we're going to be dealing with here today. And so on my pipette here, we have this little sort of angle coming off the end here. That's essentially a place to rest your hands. And that will allow you to sort of wrap your front 4 fingers around the pipette and leave your thumb, and only your thumb, to push this little button up here on top. And essentially the way that pipettes work is they have a little space inside of the pipette. And when you push on this button up here, you push down on a plunger inside of the pipette that pushes out air or liquid or whatever inside of the pipette. There's a little box on the side of each of the pipettes that tell you how much volume or how much space is actually in the pipette at a given time. So on this gray pipette here right now, I have the numbers 1, 0, 0, 0. And there's a little line separating the first 0 from the other 2. I don't know if you guys can see that there. And so what that's telling me is that I have 10 microliters of volume inside of this pipette right now. So if I go ahead and hold my pipette and I put my thumb up on this little button up here and I push down on the pipette, that's going to push out 10 microliters of air. So therefore, if I took this pipette and I put it into a liquid and then I let go, I would take up 10 microliters of liquid, thus allowing me to accurately measure 10 microliters or whatever volume it is that I'm trying to measure. So now with the plunger, with the button up on top here, when I push down, or if you guys ever have had the opportunity to work with a pipette, you may have pushed down on this button here, and you'll notice that there are actually 2 different places that you can stop the button. So if you push really, really gently, you'll encounter a little bit of resistance. We call that the first stop. So that actually pushes out the exact volume of air that the pipette is set for, that those 4 numbers on the outside are set for. If I push a little bit harder, I can push the button all the way down. So we call that second stop. That pushes out a little bit more air than whatever the pipette is set for. So they have 2 different functions when we're working with a pipette. So the first stop is primarily just for measuring. So because we want to be precise, if we set the numbers for a certain for a certain value, we can push down to the first stop and we can take up a liquid, and we can be confident in the volume that we've taken up, or the volume of liquid that we've taken up. The second stop has a little bit of a different purpose. That's essentially there to help push every last drop of liquid out of the pipette when you're sort of transferring liquids from one place to another. And the reason that we need that second stop is because most solutions in a molecular biology lab, the solvent, or the thing that they're dissolved in, are usually water. And as many of you guys may know, water has some very special properties. The water molecules like to stick to each other, and they like to stick to the walls of the container that they find themselves in. So those are the sort of properties of cohesion and adhesion, respectively. So we need that second stop just to sort of get the rest of the solution out of our pipette. But anyway, we are going to be measuring with the gray pipette. And so those 4 numbers on the outside, they range between 0.5 and 10. So for this first part of the experiment here, we're actually going to be setting up the following values. We're actually going to be setting up the experiment that you see in the table below down here. So all of the numbers in the table represent measurements in microliters. Okay, so notice that for up on the top of the slide here, you'll see all the 4 tubes that we just labeled. On the bottom, we have the different tubes, and each row tells you what's going to be going into each of the tubes in microliters. So notice that all 4 tubes are going to get buffer, all 4 tubes are going to get DNA, and then each of the tubes are going to get their respective enzyme. So the E tube only gets the eco, the H tube only gets the Hindi, the water only goes into the control, and our mystery enzyme goes into the question mark tube. So if we wanted to set our volume for 5 microliters, we take a look at our pipette here, and you'll notice there's like this little safe knob thing right here that allows you to accurately adjust the volume. So I'm going to go ahead and turn this knob clockwise. You'll notice that the numbers go down, and the little button up on top here also goes down. I'm going to do that until I get to 0500, 0500. So that little line in the middle of the pipette, in the middle of the numbers there, that represents the decimal mark. So when you're using a gray pipette or a smaller pipette, set your numbers around those values. Okay, so let's jump into setting up our digest tubes here and measuring liquid. I'm going to turn to this table here, and I'm going to go ahead and just basically go through that table and combine everything that I need into each of the respective tubes. So logically speaking, we have buffer set. We have buffer, 5 microliters of buffer rather, going into all 4 tubes. So I'm going to start there just because it's the biggest volume, and I can more quickly sort of adjust my volume down to 4 and then to 1 after that. So just sort of a workflow sort of idea, okay? So I'm gonna check to make sure that my pipette is set for 0500, and I happen to have all of the different reagents that I need right up here in this test tube rack. And I'm gonna double check the labels just to make sure that I have everything that I need. And so I take up this blue tube here, and I can see that I have the eco enzyme. I have a green tube here that has the Hindi enzyme in it. I have a clear tube that has our buffer right here. And I have a DNA tube and a DNA sample in a yellow tube up here. And then I have my mystery also in a clear tube. All right, so I'm going to go ahead and go through the measurements of each of these. So when we're dealing with solutions in a chemistry lab, we can't just use the pipette just like this. We don't want to sort of damage the inside of the pipette and contaminate future experiments by putting random chemicals inside of this pipette. So thankfully, they've come up with this nifty little invention, pipette tips. So pipette tips are designed to specifically fit on each of the different size of pipettes. So I'm going to use this box of red tips here because they have the tips for the gray pipette. And notice that the box is closed with a little plastic lid here. So the idea is that these tips are protected from the air and from contaminants that might be sort of falling in, or as I'm talking, I'm blowing air onto the pipette tips or whatever, and it might be contaminating the tips. So whenever you're working with pipettes, you want to make sure that your box of tips is closed when they're not in use. You don't want to ruin future experiments by just sort of by accident, really. Okay, so I'm going to go ahead and open my box of tips here, and then I'm going to take my pipette and very carefully just press it into one of the tips. So it doesn't take much force to get it on there. So I'm just going to basically go like that, tap it a couple times, and when I pull my pipette out, the tip is securely on there. Okay, so now I'm going to go ahead and start with my buffer like we talked about before. So my volume is set for 5 microliters. I'm going to take my buffer tube and I'm going to pull it up to my face like this. And you'll see me measuring multiple times throughout the day. And when I measure, I always hold the tube right up to my face like this. And it may seem kind of silly, but it's for a good reason. You want to make sure that you see what you're doing and you actually can confirm that you're taking up liquids and your pipette tip is where it needs to be. So I'm always going to hold my tubes and measure with it up to my face like this. All right, so I'm going to take my pipette, and to get my buffer, to measure my buffer, I'm going to push my thumb on this little button up here, and that will, I'm going to push to the first stop. That's going to push out 5 microliters of air. Okay, so when I take that pipette tip, I'm going to go into the liquid at the bottom of the tube, and then I'm going to very slowly release my thumb. That's going to take up 5 microliters of buffer. Okay, so I don't know if you guys can see that in the little tip of the pipette here, but there is 5 microliters in the tip of the pipette. Then I'm going to go to my first tube. I'm going to basically go in order. It kind of helps to be organized about this. You don't want to put stuff in random tubes and you'll forget what you added, and then it's not going to work out well for your results. So I'm going to start with my E tube. I'm going to pick up my E tube and I'm going to hold it up to my face again, and I'm going to take this pipette, the pipette tip, and I'm going to go all the way down to the bottom of the tube. Notice that I'm not going in there and just sort of trying to drop the liquid in. It's a very small amount of liquid, remember. It's probably not going to just fall into the tube. So you want to go all the way down to the bottom and gently touch the bottom of the tube. And then we're going to push to our second stop. Okay, now I'm pushing to my second stop because I'm trying to push out all of the liquid. Okay, so once I've pushed out all of the liquid, I keep my thumb on the button up top here, and then I pull out of the tube completely. Once I'm clear of the tube, I can let go of the button. Okay, so if I just went in there and I pushed to my first stop, my second stop rather, I pushed out all the liquid and then I let go, I would just suck the liquid back up again. So you want to make sure that you're doing that properly whenever you measure. So now, since I'm just putting buffer into all of these tubes, I don't have to go ahead and change my tips yet. Okay, so I'm going to keep this tip on for the moment. So now I'm going to go to my H tube and I'm going to repeat the process. So I take my buffer, go to my first stop, I go down underneath the liquid, I take up 5 microliters of liquid, and then I go over to my H tube, go all the way down to my second stop, then I let go once I'm clear of the tube. Okay, and then I'll repeat the process. Whoop. I'm going to repeat the process for my minus tube as well. So I go first stop, take up the solution, go down to the bottom, and I finally finish off with my question mark tube. Okay, so now that my buffer's all in the proper tubes, I can go ahead and get rid of the buffer, the tube that the buffer originally came from. So on the table here, I have like a little beaker. And this is basically going to be for my science waste throughout the day. So all of my empty tubes, all of my pipette tips, whatever that's like science-y, that's going to go in that garbage. Okay, so I'm going to go ahead and I'm going to change my pipette tip now because I'm going to move on to the DNA. To change your pipette tip, you're going to go ahead and take the pipette, and you'll see this little gray button on the other side of the pipette here. That will push the pipette tip right off, and you can throw it into the waste. Okay, so then I'm going to move on to my DNA next. So the DNA, remember, we need to add 4 microliters of DNA to all 4 tubes. So I'm going to adjust the volume on the pipette down to 0.400. I'm going to get my pipette tip on the same exact way. And then I'm going to go through and add my DNA from my yellow tube here to each of the 4 tubes. So again, first stop, go under the liquid, take up the liquid, and then I go to my E tube. I go all the way down to the bottom. And now when I put my DNA into this tube, since there's already liquid in here, it's best to basically inject that liquid into the DNA, that is, into the liquid that's already present in the tube. So I'm going to go all the way down to the bottom and inject that DNA into the liquid that's already on the bottom of the tube. Once again, I'm keeping my thumb on the button till I pull out, and then I can let go. So now you might ask yourself, like, I have buffer in all 4 tubes, and now I'm adding DNA into all 4 tubes, but do I have to change my tip in between? So honestly, not really, just because all of the tubes essentially have buffer and DNA, and the sort of little tiny bit that sort of gets exchanged in between is not really going to throw off the concentrations too much. So therefore, I'm not going to bother changing my tips just yet. But now that I have all of the DNA into my 4 tubes, I'm going to throw away the DNA tube. Now it becomes important to really, really change your tips. So I'm going to adjust my volume down to 1, and I'm going to go ahead and add 1 microliter of enzyme to each of my tubes. So I'm going to go ahead and start with eco and add my 1 microliter. And again, this is all the same exact process as you've seen before. But now I'm going to be changing my tips every single time. It's very, very important to change your tips every time once you've started adding enzymes. If even a little bit of enzyme gets into the wrong tube, you're going to wind up essentially contaminating your experiment, and you won't be able to tell what your mystery enzyme is. All right, so I'm adding my water to my control tube. And then I'm adding my mystery to my question mark tube. And we're done. Okay, so now, if you've done everything correctly, you should basically have about 10 microliters in each of the 4 tubes. So the next step is to take our 4 tubes and put them in our 37-degree control heat bath. So I'm going to go ahead over here. I have a heat bath or water bath. It just has some water in it set for 37 degrees Celsius. And there's a little piece of foam in here. And I'm basically going to just insert my enzymes right into the heat bath like so and close that up. And we're going to let that sit for a little bit for now. And in the meantime, I want to set up the part of our experiment. So the technique that we use to actually visualize our DNA is called electrophoresis. Okay, so you guys saw that the liquids that I was using, first of all, they're clear, and DNA is very, very small. You can't just see it with the naked eye. So we need to figure out some way of taking a look at all of our DNA at the end of the experiment. So gel electrophoresis is a pretty cool technique. It was actually invented here at Cold Spring Harbor Laboratories. And so gel electrophoresis uses one of these little chambers called a gel electrophoresis chamber. And we hook this into a power supply. The power supply is right here. And this power supply is going to basically generate an electric field in this box here. And it's going to pull charged particles, such as DNA, through that electric field. And it's going to allow it to sort of separate those particles out based on their size. Okay, so the gel part of gel electrophoresis is called agarose. So I have a bottle of agarose right over here. And agarose is, when you're making it, is actually pretty hot. So it's a liquid right now. It starts off as a powder. We mix it with a chemical called a buffer, and we make this sort of, and we heat it up in the microwave, and it makes this sort of liquid agarose. Okay, so we need to essentially pour our agarose into a sort of mold in order to sort of make basically fancy science jello, more or less. And so I have a casting tray right here. Okay, so notice that there's a black and a red line on it, but the ends are open, which may seem sort of counterintuitive for a jello mold. However, we do have a way to deal with that. So I have these little blue things right here. These are going to essentially close up the ends of the casting tray so that the gel doesn't leak out. Next, what we need to do is create places that we can put our samples in the gel. So you'll notice that there are, I don't know if you guys can see this, but there are these little sort of slits in the sides of the casting tray here. Those are designed to fit this thing. This is called a comb. And you'll notice that the little teeth on the comb, if you put those in there and then you pour gel on top, it's going to make these little sort of cups or depressions in the gel. Those are called wells, and that's basically where we're going to be loading our DNA samples. Okay, so I'm going to take that comb and I'm going to place it right here at the black line. Once we're all ready, I'm going to go ahead and pour my hot agarose right into the casting tray here, and I'm going to be careful to only pour it so that it goes halfway up the teeth on the comb. So if you guys can take a look at this, you'll notice that there's a black line on the gel. On the comb. We're basically going to pour just enough gel to get to that black line. So basically about halfway up the teeth on the comb. We don't want to go too far because if we pour too much gel, it's going to basically create one gigantic trench where the comb is, and then we're not going to be able to separate our samples. But there you go. There's our gel. So it'll take about 10 minutes or so for that gel to solidify, but I went ahead and already poured a gel for you guys, so it's right up here. So I'm going to take those blue ends off very carefully. And voila. There you go. We have a nice little gel. So if you look directly down on it, you'll see that there are 8 little wells very clearly indicated in the gel there. All right. So next, we have to set up our gel in our gel chamber and our power supply. So I'm going to go ahead and take the lid off of the gel box here. Notice that there's a black and a red line on the gel box as well as the casting tray. So I'm going to essentially lower my casting tray, gel and all, right into the gel chamber. What was it? 45. I'm going to basically put it right there in the middle of the gel chamber. And so now I can essentially prepare to hook it up to our power supply here. So the power supply has a bunch of little ports in it, and you'll notice that there's a black and a red wire. We're going to match up black to black and red to red on one of these ports here and plug it in just to get ready. The next thing we need to do is figure out if we need to add anything else. Now remember, we're trying to conduct an electric field through this box, right? So air by itself is really not a good conductor. Electricity doesn't really flow so well just through the air. We might need something like a liquid, or better yet, a solution of ions to help conduct that electric field. So it just so happens that I have a beaker filled with TBE buffer over here. And this time it's a buffer, but it's not really helping with pH so much. It's really just a solution of ions to help conduct current through the box. So I'm going to go ahead and pour just enough buffer into the box here to just cover the top of the gel. Okay, so I'm going to start on one side. I pour enough buffer for it to go over the top of the gel. Then I'm going to go to the other side and pour a little bit more buffer, and it's going to just go ahead and cover the wells. Okay, so now the wells that you used to be able to see from sort of the side, now you can't really see it as easily. You have to almost look directly down at the wells. Okay, so now that our gel is all sort of set up and ready to go, let's go back to our samples. Okay, so remember that our samples are clear. So you can't really see them, but with even your eye. So if we put them into that box here and load them into the gel, we're not really going to be able to see anything. We need to add something that will help sort of give color, weight, and also stain for the DNA. And we're going to use something called cyber green. So I have a tube of that right up here. So it's a pretty thick, viscous sort of dye. And cyber green does multiple things. It helps to give color to our sample for us to see it. It helps to weigh down our sample in the gel so that the DNA doesn't float away. And it also has this cyber green stain in it, which will bind to the DNA and allow it to be visualized underneath a black light. We'll actually be able to see it fluoresce. So we're going to add 2 microliters of cyber green to each of our digests. So it's been about 10 minutes or so. That should be enough time. So I'm going to go ahead and take my digests out of the heat bath here. And now I'm going to add 2 microliters of cyber green to each one of my digests. So this is carried out the same exact way that we did earlier. So I'm going to basically take my cyber green tube, and by the way, cyber green is pretty viscous and nasty, so when you're working with this, you want to be careful. If it gets on your clothes, it'll damage your clothes, and if it stays on your skin, you're going to be blue for a while. But, that's why I wear blue. So I'm going to go ahead and measure out 2 microliters of cyber green. I'm going to insert it right into the liquid in my E-tube. I'm going to do the same thing for the Hindi tube. Now, question, do we need to change our tips now? Yeah, we do, right? If we touch this tip to the eco solution and then we touch it to the Hindi solution, we might contaminate our samples. We don't want to do that. So I'm going to go ahead and I'm going to go ahead and take the tip off and get a fresh tip every single time that I am adding dye to a new tube, to a new digest tube. Okay, so I'm going to go ahead. Do the same thing. And I'm going to do that for the minus tube and the question mark tube as well. Okay. So once we've added our dye, you'll notice that the clear solution, or what used to be a clear solution, is now blue. It actually has some color to it, so we can actually see it now. So check that out. You can actually see that the liquid is now blue. This will help when we're trying to load our samples into the gel. So let's go ahead and get that taken care of. So we're going to load 10 microliters of our samples into the middle 4 lanes of our gel. So if you look up on the board here real quick, essentially this, we're going to basically skip these left and right lanes, and we're going to basically load E, H, minus, and question mark, just like that, into the middle of the gel. And the reason for that is because they sort of flow, the sort of DNA is going to flow through the gel a little bit more evenly in the middle of the gel, and it's just a little more organized for us to visualize. So now, I go to my gray pipette, turn up, I'm going to basically adjust the volume all the way up to 10,000 for 10 microliters. And making sure that I'm changing tips every time, I'm going to go ahead and add 10 microliters of each of my samples to the respective lanes. So I'm going to start with my E tube here. Okay, I take my 10 microliters. That tube is garbage at this point. And when you're loading a gel, it's a pretty difficult thing to do, especially if you've never done it before. It's all about stability. So I drink a lot of coffee, so I shake a lot. So my hand is already shaking like right now. And so I have to make sure that I'm being very stable in the way that I do this. So what I like to do is put my elbow on the table, stabilizing my hand, and then I use the other hand to guide the tip of the pipette right into the well where your sample needs to go. So if you take the tip of the pipette and just go right beneath the surface of the liquid into the well, remember you're aiming for the well, you should already be in the well if you've basically added enough buffer just to cover the surface of the gel. So I'm going to go in just a little bit, and now I move the tip of the pipette sort of up and down a little bit, and I can basically verify that I'm actually in the gel. When we dispense our sample, when we actually put our sample into the gel, we have to make sure that we're not going too quickly with this. We don't want to blast the sample into the well and then up out of the well and into the buffer. It'll float away and you'll never see it again. So we want to go slow with this. So I'm going to go into the well, and I'm going to basically go to my first stop only when I dispense the sample, when I push out the sample. The reason for that is because if I go to the second stop, I'm also going to push a little bit of extra air in there, which is going to sort of throw off the position of the sample. It's going to blow it out of the well, or it might anyway. All right. So once I'm done with that, I keep my thumb on the button and on the plunger on the end of the pipette, and then I can let go once I'm clear of the gel. Now, if I've done everything correctly, I should see a little blue rectangle where the well is. And that's to be expected, right? Because the comb that we used has sort of like these little rectangular teeth. So our wells are all going to look like rectangles. Okay. I'm going to go ahead and do the same thing for each of the other tubes. Okay, so I go to my H tube, my H lane, and I go ahead and dispense my H into that lane. Then I go ahead to my next tube. Hang on, let me just get a fresh thing of tips here, guys. Give me 1 second. So these tips are pretty nifty. We have this sort of interchangeable box here. I can pull out the old tray, and then I can replace it with a new tray right here. And it has a little sterile lid just to keep everything sterile. All right, so I'm going to go ahead and load my last 2 samples now. So here's my minus tube. And last but not least, here is my question mark tube. Okay. All right, we're all done. So we have our samples loaded nicely in there, 4 little rectangles, and they look pretty good. So I'm going to go ahead and close the gel box by sliding these little poles back on the electrodes. I'm going to turn on the gel box, and I'm going to set it for 140 volts. So when you turn on the gel box, you'll notice that it defaults to 0 volts. And we can use the up and down arrows to essentially set the amount of voltage. And voltage, by the way, is a measure of the electrical potential energy. So it's basically the amount of electrical charge that you want to be conducted through the box. So you have to be careful when you set this, because you don't want to roast your samples. You don't want to make it too high, but you want to give it enough of a strong field so that we can efficiently pull our DNA through the box. Then I'm going to set it for 45 minutes. We're going to let that sit and run for 45 minutes. And don't worry, guys, I already have a gel prepared for you guys. We're going to take a look at that in just a few moments. Okay, there we go. Then I'm going to go ahead and push this little running man start. And you guys may not be able to see this, but if you look into the gel box, into the buffer that I poured, on each end, you'll notice that there are these little wires and there are little soda fizzy bubbles coming up off of the wire. And that just indicates that there is a current flowing through the box. So let's let that do its thing. In the meantime, though, I've already prepared a gel for you guys using the same mystery restriction enzyme. So let's take a look at what we should see. So this little machine here is a little sort of handheld illuminator, and it sends this. Oh, actually, I wanted to show you first. I don't know if you guys can see this, but there is a blue and a purple line on the gel. I don't know if you guys can see that. So that blue and purple line essentially is the dye that we loaded into the gel previously. So one of the other functions of that loading dye is it tells you just how far or how long your gel has been running for. And so the dye that I put into our tubes actually had 2 different components to it. One that runs a little bit faster, or a lot faster, I should say, than the other. That's that purple one down there. And then there's one that runs a little bit slower. That's that blue one up there. And that just basically tells you that the gel went for a certain amount of distance, or a certain amount of time, I should say. Okay, but often students, when they do this lab at the DNA Learning Center, will take a look at this and be like, oh, I didn't do anything right. I can't see any DNA. But remember, cyber green, that stain that we used, can only be visualized with a black light, with a UV light. So we have to use one of these UV black lights in order to actually see our DNA. So I'm going to use this handheld illuminator and see if we're able to see . I don't know if we really can here. Might have to look at this up on the board. I don't know. Can you guys see that? Sorry. Can you guys see that? Yeah, sorry, guys. It's a little bright in this room here, so it's difficult to see the results. Oh, that works. You guys able to see that? They kind of can. Basically, the cool thing about this little transilluminator is when the DNA actually fluoresces under a black light, it's much cooler to see than on our camera up on the board here. But I'm going to go ahead and project this up on the camera just because we can sort of see everything a little bit better. I don't think you're going to be able to tell me what the mystery enzyme is based on, oops, sorry guys, based on the based on this. So let me go ahead and bring this over to our camera up at the front of the room here. Okay. So it's a little faint. And also, I loaded everything out of order. So I got to be honest with you guys, right before this lab started, I started a gel, and then the mystery enzyme didn't work. In about 10 minutes, set up another gel. So I think I misloaded them, or I loaded them out of order. But whatever, we can still answer our question. So up at the front of the room here, you guys may be able to see our DNA. Our samples. So the first well is the first sort of lane here is our E lane. Oh, well, okay, that's our E lane. This is our H lane, and then I just switched around the minus and the question mark. But the idea is still there. So if we take a look, what we see are all of these different fragments. And so the purpose, or what gel electrophoresis actually does, is it separates out DNA on the basis of its size. So if you remember those restriction maps that we showed you before, you'll notice that there were various different sizes of fragments. Okay, I'm going to show you in a moment that'll sort of solidify that point. But in fact, let me jump to that right now, actually. So here's our restriction maps again, and I'm going to zoom in here. And at the eco restriction map, you should expect to see 1, 2, 3, 4, 5, 6 different bands for eco. And notice that there's a really big one, 26 base pairs, and there's a couple of other fragments in 3,500, 500, 5,800, et cetera. For Hindi, we have a similar sort of idea, except here we have 1, 2, 3, 4, 5, 6, 7, 8 different fragments. So if we take a look back at our results here, notice that for the eco lane, that's this first lane on your right, the eco lane has, it's a little bit sort of light and faint there, but you can just barely make out 1, 2, 3, 4, 5 different bands. If we take a look at the second lane, that's the Hindi lane, we can make out 1, 2, 3, 4, 5, 6 different bands. So it's much less than what we expect. Actually, I'm sorry, for the Hindi lane, we can actually make out 7 lanes. If you guys look all the way down on the bottom, you should be able to see one very, very faint lane on, like right down here. I don't know if you guys can see that, but it's, if you guys see those, like right between the bubbles. Just look between the bubbles, you'll see them. So there's 2 little faint bands right down here. So we have 7 different bands, okay? Now, I sort of gave it away, but if we look at the next lane, that's our mystery. What do you think our mystery enzyme is based on the cut patterns? Right? It matches the Hindi, right? So therefore, my mystery enzyme is Hindi, for sure. Okay, the control all the way over here, notice that there is no multiple different banding. It's actually just a single band, and that indicates that there was no cutting going on. That's what we should expect to see from our negative control. But why do we see why do we only see 5 bands for eco and 7 bands for Hindi, though? So as it turns out, let's look at an ideal gel over here. So here we have our eco, and here we have our Hindi. Why do you think we only see 5 bands for the eco? And if you take a close look at the restriction map, you'll notice that 2 of the bands, specifically 5600 and 5800 over here, are very, very close in size. So if we look at our sort of gel results here, I can move away this little bar here, and it will show you what each of these different bands are supposed to represent. So essentially, one of the bands, the 5800 and the 5600 band, migrated together. They were so close together that you couldn't tell them apart. For the Hindi, one of our bands is very, very small. There's actually one that's about 125 base pairs in size. It's very, very small. That's probably so small that it migrated all the way off the gel and out into the buffer. So basically, one of the Hindi bands is missing. You can't really see that. And this sort of illustrates the idea that the smaller the fragment of DNA, the farther it's going to wind up going in the gel. It's kind of like traffic on the expressway, on a highway, major highway. You can picture like a little fragment of DNA being like a motorcycle. They have a really, really easy time of getting through all the traffic. Just like a little piece of DNA is going to have a really easy time navigating through the gel. A bigger piece of DNA is kind of like a coach bus or a tractor trailer. It's not going to have an easy time getting through the gel, and it's going to wind up getting stuck closer up here to the wells. Notice, guys, too, that on the gel, for the gel box, at the top of the gel here, we had to make sure that there was a difference in charge from one side of the gel box to the other. So when we set up the gel box, the black electrode corresponds to the negative charge, and the red electrode corresponds to the positive charge. And that we do that is because we load our DNA up at the black line, up by the anode, right, by the negatively charged electrode. That's the negatively charged DNA through the gel and pull it towards the positively charged electrode. Sort of like that. All right, so hopefully you guys had fun today. We did answer our question. So remember that mystery enzyme wound up being Hindi just because it had a similar cut pattern to the Hindi control. And if we were to repeat this experiment using a different mystery enzyme, we should be able to figure that out as well, just by matching it to our eco and our Hindi cut patterns. Hope you guys had a wonderful time. Stay safe out there and enjoy the rest of your day.